Complex lipids are lipids that are bonded to other types of molecules. The most common and important complex lipids are plasma lipoproteins, which are responsible for the transport of other lipids in the body. Lipids are only sparingly soluble in water, and the movement of lipids from one organ to another through the bloodstream requires a transport system that uses plasma lipoproteins. Lipoprotein particles are spheres that consist of a core of hydrophobic lipids surrounded by an outer layer or a shell of amphipathic proteins, phospholipids, and cholesterol. As depicted in the picture shown down here at the bottom right, the amphipathic molecules of the outer surface are able to interact with the aqueous environment of the bloodstream, and the hydrophobic molecules in the interior of the lipoprotein particle. And so we have all of our hydrophobic molecules on the interior, and then we have our amphipathic molecules that make up the shell or the layer that the outer layer of the shell, I should say. And on the outside, we have all the portions that are going to interact with the aqueous phase, so we have all the hydrophilic portions. And then on the interior, on this part here, we'd have all the hydrophobic portions. There are four major classes of human plasma lipoproteins: chylomicrons, VLDL or very low density lipoproteins, LDL or low density lipoproteins, and HDL or high density lipoproteins. Chylomicrons, which have a density of less than 0.95 grams per milliliter, carry dietary glycerides from the intestine to other tissues. The remaining lipoproteins are classified by their densities. VLDL, or very low density lipoprotein, has a density of 0.95 to 1.019 grams per milliliter. They bind to triglycerides synthesized in the liver and carry them to adipose and other tissues for storage. LDL, or low-density lipoproteins, are characterized by the density of 1.019 to 1.063 grams per milliliter. They carry cholesterol from the liver to peripheral tissues and help regulate cholesterol levels in those tissues. These are the richest in cholesterol, frequently carrying 40% of the plasma cholesterol. Since they carry so much cholesterol, 40%, this is why we call LDL the bad cholesterol. HDL, or high-density lipoproteins, have a density ranging from 1.063 to 1.210 grams per milliliter. They are bound to plasma cholesterol, however, they transport cholesterol from the peripheral tissues to the liver. Low-density lipoproteins deposit cholesterol on the walls of arteries when they carry more than is needed from cell membranes. And this is why a diet that's high in cholesterol can lead to plaque, which restricts blood flow. Thus, LDL we call bad cholesterol. HDL, or high-density lipoprotein, reduces the level of cholesterol in the bloodstream by bringing the excess back to the liver where it can be metabolized. HDL is what we call the good cholesterol. Chylomicrons are aggregates of triglycerides and protein that transport dietary triglycerides to the cells throughout the body. Not all lipids in the blood are derived directly from the diet. Triglycerides and cholesterol are also synthesized in the liver and transported through the blood in lipoprotein packages. Triglycerides synthesized in the liver are assembled into VLDL particles that carry the energy-rich lipid molecules either to tissues requiring an energy source or to adipose tissue for storage. Similarly, cholesterol synthesized in the liver is assembled into LDL particles for transport from the liver to peripheral tissues. Entry of LDL particles into the cell is dependent on a specific recognition event and binding between the LDL particle and a protein receptor embedded within the membrane. The molecules on the surface of the LDL that binds to the LDL receptor is the protein apolipoprotein B. There is one apolipoprotein B molecule on each LDL particle. Low-density lipoprotein receptors, or LDL receptors, are found in the membranes of cells outside the liver and are responsible for the uptake of cholesterol by the cells of various tissues. The apolipoprotein B protein on the LDL particle binds specifically to the LDL receptor, and the complex is taken into the cell by a process called receptor-mediated endocytosis. The membrane begins to be pulled into the cell at the site of the LDL receptor complexes. This draws the entire LDL particle into the cell. Eventually, the portion of the membrane surrounding the LDL particle pinches away from the cell membrane and forms a membrane around the LDL particles. Thus, they can form a vesicle or endosome containing the LDL particles. Cellular digestive organelles known as lysosomes fuse with the endosomes. This fusion is accomplished when the membranes of the endosome and the lysosome flow together to create one larger membrane-bound body or vesicle. Hydrolytic enzymes from the lysosome then digest the entire complex to release cholesterol into the cytoplasm of the cell. There, cholesterol inhibits its own biosynthesis and activates an enzyme that stores cholesterol in cholesterol ester droplets. High concentrations of cholesterol inside the cell also inhibit the synthesis of LDL receptors to ensure that the cell will not take up too much cholesterol. People who have a genetic defect in the gene coding for the LDL receptor do not take up as much cholesterol. As a result, they accumulate LDL cholesterol in the plasma. This excess plasma cholesterol is then deposited on the artery walls, causing atherosclerosis. This disease is called hypercholesterolemia. There's also some evidence that HDL in the blood helps reduce the incidence of atherosclerosis, perhaps because HDL carries cholesterol from the peripheral tissues back to the liver. In the liver, some of the cholesterol is used for bile synthesis and secreted into the intestines, from which it is excreted. In one study, the benefits of HDL extended until a level of 90 milligrams per deciliter was reached, and then the effect leveled off. Further research is needed to understand the extent to which HDL provides protection from heart disease. A final correlation has been made between diet and atherosclerosis. People whose diet is high in saturated fats tend to have high levels of cholesterol in the blood. Although the relationship between saturated fatty acids and cholesterol metabolism is unclear, it is known that a diet rich in unsaturated fats results in decreased cholesterol levels. In fact, the use of unsaturated fat in the diet results in a decrease in the level of LDL and an increase in the level of HDL. With the positive correlation between heart disease and high cholesterol levels, the current dietary recommendations include a diet that is low in fat and the substitution of unsaturated fats or vegetable oils for saturated fats, animal fats.